Hello friends, let us talk about one of the deadliest disease ever known to human beings. So this is one of the greatest damage which is done to particularly countries of West Africa. You might have heard about this disease. This disease is called Ebola. And Ebola outbreak is happened in the West Africa. And we are going to discuss what was the crisis it faced, what was the response, and what is the lessons we learned. And initially, we will look at what is this Ebola and what do you mean by Ebola viral disease and what is Ebola virus, how it looks, what is the structure of that, then transmission, then what are the symptoms? Is it similar to normal virus or it shows some other symptoms? Then how to contain it or treatment it? What is the possibility? What is the technology here? Then we will look at the contextualized way that Ebola outbreak in West Africa. So what exactly they've done to contain it? What was the response of World Health Organization? How they manage with the technology? So we are looking at this one in all the scenario of Ebola outbreak in West Africa. First, we will look at Ebola viral disease and what do you mean by Ebola virus. This EVD or Ebola viral disease, formerly known as Ebola hemorrhagic fever, is a severe and often fatal illness in human beings. And this disease is caused by Ebola virus, a member of the Filoviridae family, which includes Ebola virus, that is Zaire Ebola virus, Sudan Ebola virus, Thai forest Ebola virus, Bundibugyo virus, that Bundibugyo Ebola virus, and Reston Ebola virus. This is the names of the scientific name as well as commonly what we call. So this is particularly this hemorrhagic fever, which mainly affect the brain and other peripheral organs. But at the same time, this is caused in a different part of the countries like Liberia, then maybe Sierra Leone, or maybe other part of the West African countries. It affected n number of people across that West Africa. As usual, if you look at any virus, it has got a specific envelope, then it's capsid, then what you are calling viral glycoproteins and all those things, and genome. Mainly RNA is the genome which you can see in Ebola virus, it's RNA virus. And this Ebola virus is like a thread-like structure. You can see, you might have seen in the earlier picture, it is looking like a thread-like structure. So it has got an envelope. The Ebola virus has an outer envelope that surrounds the viral particle, and this envelope is derived from the host cell membrane during the process of viral budding. As you in earlier chapter we discussed that when a virus replicates, it takes everything from a host. So that means if a Ebola virus is having an envelope, that envelope is derived from the host cell membrane. Next is viral glycoprotein. The viral envelopes contain glycoprotein spikes, which are crucial for the virus to attach to and enter host cells. So that's important thing. There will be some key in virus. So this key, if it matches with a lock and a cell, so it can open up the cell and enter inside. If this is not matching, then it cannot. So that's why these viral glycoproteins are important. They act as a key, so it can bind to our receptors on cell membrane surface, and it can open up the cell and enter inside. The glycoprotein is the largest target of neutralizing antibodies and is a key component development of vaccines. So whenever we develop vaccines or whenever our body produce combating machineries called as antibodies, they target the cell by looking at these glycoproteins. These glycoproteins are highly specific. Ebola viral glycoprotein is specific to Ebola virus. You cannot find them in any other virus of other categories. There may be another kind of glycoprotein. So for us, one thing is this glycoprotein is important. If we know clearly about this glycoprotein, we can develop a techniques to manage this virus. The nucleocapsid. Inside the viral envelope, the Ebola virus has a nucleocapsid core, and the nucleocapsid is composed of viral RNA and associated viral proteins. So here the important thing is viral RNA. So the Ebola virus has a single stranded negative sense RNA genome, and the genome encodes the viral protein necessary for replication and structural protein that form the virus particle. So it forms all the protein which are necessary for this virus, especially Ebola virus, to emerge from a host cells and continue its infectious cycle. So it need a specific single stranded RNA which codes all the genome which is required for this Ebola virus to survive in any hostile conditions or maybe in any host. So the Ebola virus also got a viral protein. The virus produced several structural and non-structural proteins during its replications. Among the structural proteins are nucleoprotein, viral protein 35, viral protein 40, glycoprotein GP, and viral protein 24. These are all important proteins which you can see in case of virus. I mean, particularly Ebola virus. GP means the glycoprotein which you can find on surface of the cell membrane. But the fun is that cell membrane, the membranous part is completely derived from the host cell. Then the matrix. The VP is a matrix protein that play a crucial role in assembly and release of viral particles. So the important thing is once a virus enters inside a cell, if its glycoprotein matches with the cell membrane, it can easily open up the cell and it can come inside. Once it is inside, it will multiply. Once it is multiplied, next duty is to release them, release the viral particles. So once the viral particles are released, that can infect the neighboring cells. So the VP40 play important role in that one. That is viral protein 40. It is evolved in the formation of viral matrix beneath the viral envelope. There is a viral matrix below the viral envelope where you can see the concentrated VP40, which will help this viral particle to assemble and then get released and continue the infection cycle. RNA-dependent RNA polymerase. The Ebola virus encodes RNA-dependent RNA polymerase, which is essential for replicating genome with the host cells. So RNA-dependent RNA polymerase is the enzyme which is responsible for the replications and all the cycles which is related to Ebola virus. So these are all the components. So first what we have seen is next we are going to see about, discuss about transmission. So this is the important phase where we need to stress upon a little bit. We have seen what is the structure of that virus. It has got a single-stranded negative, antisense DNA, and in the RNA, which play important role in its multiplication and other things, as we all know about it. So how it transmits? So the important thing is in this modern world, we are in a continuous contact and our human population is too high. So for this virus, it is easy to get a host. From one host to another host, it can easily jump, which wasn't possible in earlier days. Getting a host was difficult for a virus in the earlier days. But now it's easy because number of people are close by and the virus can easily attack us. So direct contact with the blood or secretions or organs or other body fluids infected people. So that means all this infected fluid which is coming out of the host body may have this virus or viral particles. This can occur through broken skins or mucous membranes such as eyes, nose, or mouth. That means even though we call it is our body is well protected, but there is a chance that if a simple droplet from, the droplet of saliva from one person to another person, which is sufficient for the transmission of this virus from one individual to another individuals. Next is direct contact with the surface and material contaminated with virus. This is the things you might have observed during COVID infection also. If a particular person touches any surface, we need to clean the surface with antiviral agents or maybe disinfectant. Otherwise, if someone else touches there, this virus will survive. So now the question is whether the virus will survive for a longer time or it's short time. That depends on virus. Sometimes this virus will get converted into the crystalline state and it can survive for a longer time. But it won't happen. So occasionally it happens. But which is also sufficient to continue the cycle. That is important, right? So for continuing the viral infectious cycle is important. So which this thing is sufficient. There is no need of a special mechanism. The virus can survive on surface for several hours to days. That depends on virus. And handling and consumption of bushmeat wild. So this is the challenging thing, particularly when there is no unorganized societies or societies not advanced societies. So particularly African society is not advanced like other world society, and they are more of traditional belief and traditional way of cooking things and eating. And the one important thing is bushmeat. Anything they will get it just maybe they hunt animal and they just dry it and eat it. They don't look at whether this one is cooked or not. They just eat this raw bushmeat which are just dried or smoked and they feel that is sufficient to kill the pathogens, but in real sense it is not. They are just acts like a reservoir of virus which is sufficient for human beings to get that infection. So that's what the challenging thing in particularly the tribal rich zone of the world like Africa, where if a particular person from outside put up a new protocol to follow, but people don't follow it mainly because of their indigenous belief. So the indigenous belief is one side, it is good, but at the same time this indigenous belief is really bad, particularly for this viral contamination or containing the viral infections and all those things. And next, human to human transmission. This is the another biggest challenge. If you touch the surface, you will get infection. If you eat a bushmeat, you will get. But you just look at a third scenario where human to human transmissions. This can occur through a close contact with the blood, secretions, organs, or other bodily fluids of infected people, as well as through contact with surface and materials contaminated with this fluid. So that is the important thing. During the COVID, we understood how to contain the disease by just cleaning the surface and all, which is not in all the cases. But in third world countries and all, it's very difficult to impose this kind of stringent law to contain the viral growth. But real sense, if some domestic animals got infected and from domestic animals, human beings can catch the disease easily. So this is the way. Then direct contact or direct contact with surface or materials or human to human transmission. These are all the different possibility of viral load transmissions. So if a particular person, individual got infected with the viral, I mean, what is the symptoms or clinical manifestation of his infections? So the incubation period for once the virus is inside, its incubation period will varies from 2 to 21 days. That means some people they should start showing the disease faster. Some people wait till 1 month to get disease or symptoms. So what are the symptoms common? That is high fever, then fatigue, muscle pain, then headaches, sore throat, fatigue, fever, migraine, headache, sore throat. This is followed by vomiting, diarrhea, rash, impaired kidney and liver functions, and in some cases internal external and the severity of the disease can vary and mortality rates can be high. Now, if you look at the Ebola virus, more than 90% mortality rate, only supportive care can be given. But there is no other options like virus, I mean the vaccine and the vaccines and other things are still under development process. So this one is contained only through proper care, and the severity of disease can be reduced through reducing the load. So now the wearing mask is important for us, that even at least we protect us from some diseases. So that is important. This is a crucial thing where a person is got infected or showing the symptoms similar to Ebola viral infections. In such a cases, how to diagnose this one? So this is the challenging thing. The diagnosis, we need a rapid, a diagnosis technique, and it should be readily available, but it is not so easy in all the cases. So here in case of Ebola, what they'll do is mainly they go for PCR. That means the first they'll collect the sample. Those samples will be put into viral transport medium. Once they put in viral transport media, they extract that, they go for isolation, serological test, and particularly they identify this virus while using RT-PCR. So this is the one more easily available technique through which you can confirm the presence of particular virus by just looking at some genetic materials. In earlier days, based on a symptom, based on a kind of behavior the virus shows in our body, by then we were managing the disease. But nowadays it is not like that. We have a readily available technique like PCR, polymerase chain reaction. So through which, by using a primer, what we can do is we can identify them easily. And one more important, it's not easily, it is readily available and can be confirmed with most of the reagent what is available in our lab. And early diagnosis is crucial. That's important. We need to diagnose early and start giving a the supportive care. One side, it can prevent the patient from collapsing or getting some collateral damage, or else it also prevents the spread of virus. Because most of the time, tracking, live tracking of a particular person's history is very difficult. He might have moved to different places, different functions, and he might have met different people. Then challenging their the challenge task is to get the informations and select the primary contact links, then secondary contact links, then tertiary contact links, list from index patients, so then we can treat them separately. Otherwise, it is very difficult to contain the disease. What are the possible options? If a particular person got infected and diagnosed as a Ebola, is there any possibility to treat? There is no specific antiviral treatment for Ebola viral disease. Supportive cares, including rehydration and treatment of specific symptoms, can improve the chance of survival. Experimental treatments and vaccines have been developed and tested, showing promising results, but their use is still under investigations. They are developed vaccines, it is under trial mode. So what they are doing is they are giving in a challenging mode. Preventive measures include hygiene practice, regular hand washing, because after corona we were little more punctual to this and we wash our hand regularly. Regular hand washing with soap and water and then use of hand sanitizers. Safe burial practice, proper handling and burial of dead. So once a particular person is got infected and died, then bury them safely. Otherwise, from there also there is a possibility of spreading of disease. Avoiding contact with the wild animals, especially in areas where Ebola is known to be present. In a particular area, there is a possibility of infections. In such a cases, we should not eat bushman's meat, or we should not in contact with the particularly what do you call blood and other things from these wild animals. So through all these ways, we can treat and prevent viral infections like Ebola. Next, we will discuss about a contextualized scenario, what really happened in West Africa. In 2014, the West Africa faced one of the most devastating public health crises in the history, the Ebola virus outbreak. Because it is the suddenly outbreak happened, they don't know much about that virus, and it will become a underdeveloped country. It is a public health challenge. This deadly virus, known for its high fatality rate and rapid transmission, we can break the havoc primarily in Sierra Leone, Liberia, and Guinea. Particularly country like Sierra Leone, Liberia, Guinea, there is no proper political stability, and primary health sector is also not much funded by the federal government. So what really happened is primary health sector is already in the mode of collapse. And the same time, suddenly a deadly viral infection starts, and it starts spreading from people to people. And there was a videos and showing the people are dying and falling and dying in the road itself. No one could touch them and don't know how to touch. If they touch, they will also get infected. So that is the biggest challenge what they were facing there when they were working with African, particular West African continents. The outbreak not only claimed thousands of lives but also exposed critical flaws in global and local health system, sparking a reevaluation of preparedness and response strategies for future pandemics. So when we look at any disease, we understand this is a disease X, so it causes, and because of that the disease X, these are all the problem appears, and we have a clear idea and strategies to manage disease. But it was not the case where the country wasn't not prepared. At the same time, the whole globe wasn't not prepared to face this critically. Deadly, highly fatalities having this disease called Ebola. Now, what is the epidemiology of the Ebola virus? The Ebola virus is a member of Filoviridae family, is responsible for severe hemorrhagic fever, as I told. The outbreak in West Africa was caused by a Zaire Ebola virus. I told that many Ebola virus. In that one, there in West Africa caused by the Zaire Ebola virus, the most virulent strain of the virus. You just imagine. There are different strains. One strain is more virulent than another strain, but here in case the most virulent strain got it. The virus is transmitted through a direct contact with the blood, secretions, organs, or other bodily fluids of infected people and the animals, and making it highly contagious. So it is one of the contagious disease whatever human seen. So one of the contagious disease, and through which it was spreading across West Africa. Initial outbreak and rapid spread. When it was the outbreak was first identified in Guinea, Equatorial Guinea, in December 2013, but went unnoticed for several months due to limited healthcare infrastructure and surveillance capacities. It was appeared there. People got infected. Many might have died, and there was no proper documentation and proper structure, so the people could not uh, get a proper information about it. By the time the World Health Organization declared a public health emergency of international concern on August 2014, the virus had already spread to Sierra Leone, Liberia, reaching unprecedented levels. So that is the thing. See, when a viral infection happens or viral outbreak happens, the only way is if there is no proper treatment, we can give supportive care. Same time, the creating awareness is very much essential, so we can contain the disease. Unless we create awareness, it is very difficult to contain the virus. So virus will keep on spread from one place to another place. This is what really happened in case of initial outbreaks. During that outbreak, what was the challenge they face? So several factors contributed to the rapid spread of Ebola in West Africa. One is weak healthcare system. So the political willpower is very low there, and the government itself is underfunded and insufficient tax collection, and they are only bribed by the biggest industries. So what really happens? They have a very weak, poor. I mean, one of the poorest healthcare system and insufficient resources. Just imagine there is a need of oxygen. If oxygen is not available, or else you don't have a facility to produce sufficient oxygen, so then it's insufficient resources. They need a doctors, they need a hygiene place to treat the patients. They were treated all in the field. Inadequate training of healthcare workers. That is one more prominent thing. What is that? Healthcare worker training. So if a healthcare worker himself or herself don't know how to treat, how to manage this disease, then how it is possible to contain the outbreak? In addition, cultural practices, traditional belief played a role in fueling the epidemics. So this is one more thing. Some culturally or traditionally, some belief says that you don't take treatment. So which will fuel the epidemic? Because as for the science, we know that some procedures and protocol put forward to deal with this kind of viruses. So if we properly follow, we can contain them. But there are cultural belief, traditional belief, indigenous belief. These beliefs will put as a barrier where you cannot think of, and which will fuel the epidemics. Some communities resisted interventions and safe burial practices. That is the one more thing. Some communities don't bury according to the international protocol, World Health Organization protocol, bury according to our traditional practice. Because of that, what happens? It spreads. The international community faced challenges in coordinating an effective response, delays in mobilizing resources, logistic difficulties, and one more thing is road and mobility. Why do we need a road and mobility? This is one challenge. If in isolated place a particular infection happens, you need infrastructure building, a structural building, then you need to mobilize the resources. If there is no proper supply, then it is very difficult. Logistic difficulties, lack of communication among affected countries hindered containment efforts. The slow response highlighted the need for more coordinated and robust global health infrastructures. So this is because of this challenge. One thing we learned, we need to have a proper coordination among different countries. Some countries are good in managing the disease. Some countries are developing their healthcare system, so they can help each other and manage this kind of challenging situations. So what really, but still there is a hope. We say every crisis there is a hope. So there was what was a humanitarian response and how international cooperation happened there. As the crisis escalated, the international community mobilized resources and personnel to provide aid to affected countries. You might have, I mean, the organizations such as Medicines Sans Frontiers or else the Doctors Without Borders. So the United Nations and various governments played a crucial role in establishing treatment centers, deploying medical teams, and implementing public health campaigns. So these are all important things. So we need some volunteer doctors. So if doctors tell that they can't treat this, they don't have idea, they need to be trained in a scientific way. Once they are trained, they need to be mobilized. You need to have a treatment centers, and same time you need to create a public awareness so the people come to the treatment center. They will be screened, then from index patient to primary contact, secondary contact, tertiary contact, like that. You can put them in isolate, observe for symptoms. If vaccines are available, we can give them vaccines. So this is a really challenge, but implementing public health campaign is very important. Because when outbreaks happens, and that too in case of such a deadly diseases, unless the people are informed about it, people are educated about particular disease, it is very difficult to tell them, very difficult to manage this kind of diseases. So now when we look at this and the present COVID 2014 to 2020, so the role of technology and research, this is important. And if you look at this Ebola disease and treatment, that many doctors from other part of the world also got infected and passed away. So that's a challenging thing. There are volunteer doctors which are designated by UN and World Health Organization. They will go and treat, and they don't get proper facility there. They don't have a hygiene toilets. They don't have a proper accommodation facilities. They have to treat them in a place wherever it is available. So it's a challenging. In quest to find a solution to Ebola crisis, the technology and research played a pivotal role. That is the important thing. Whenever problem happens, the people start thinking about it. how can we manage this? How well we can manage so that this problem won't appear again. So technology is the one which helps us a lot. Rapid diagnostic tests. That means how rapidly we can diagnose the things, the way we can prevent it, further spread. Experimental treatments. That means we wanted to develop early interventions. So by taking some experimental animal and treat them and get kind of information of the basics, so then the trial run we can give that medicine to people. The vaccine development efforts were accelerated. This is one more thing. The technology helped us to accelerate the vaccine development. Infection has happened. Maybe within 1 or 2 months we could develop a vaccine. Then it is easy. The use of mobile technology for contact tracing. That is a one more important thing. By using telecommunication, mobile communication system, we can trace the contacts. Public health messaging also provide instrumental in controlling the spread of virus. That means a particular person is got infected. If you wanted to send a mass message to a communities to keep them, those who are contacted that person in isolation mode, so we can send a mass message. So through which also we can manage. So that is the interesting things. Now learn the lessons and strengthened global preparedness. So this is the one thing what we learned from Ebola outbreak in West Africa. The Ebola outbreak underscored the importance of building resilient and the response healthcare systems in vulnerable regions. So that means when we think about Ebola in underdeveloped countries, how good the infrastructure is. So that's important that the development of new healthcare policies and new healthcare, I mean, the building which are dedicated only for this purpose. It has prompted a re-evaluation of global health governance, highlighting the need for improved coordinations, communications, resource mobilization during emergency situations. The World Health Organization is the one agency which manages all this, but they got a limitations because these World Health Organizations are funded by some of the developed countries. If some fund shortages are there, if a developed countries are not ready to mobilize their resources, that's why the global coordination is very much essential in such emergency situations. The World Health Organization reform and the establishment of the Global Health Security Agenda, GHSA we call, were direct response to the shortcoming revealed during Ebola outbreak. That means what happens is every year we keep on reform our policies. As we reform policies, there is a possibility for flaws. These initiatives aimed at enhance the international cooperations, strengthen surveillances, response capacities, promote research, development of medical countermeasures. So this measuring, I mean, the developing a countermeasure is also important because particularly West African countries, they are poor, their economic situation is really bad. So how they cannot have a localized countermeasures. So that some well-developed countries, those who are funded well by their federal ministries, what they can do, they can support this kind of countries. So it's because one day this simple problem of West Africa can be a problem of globe. To avoid that in the early stage, we can manage this. Once we manage, see now it was happened in there, they manage somehow they managed to contain the disease. The world is not facing the problem. But the same problem, same thing wasn't happened in case of Wuhan virus, that is coronavirus. We could not contain at the initial level. They kept it in a secret mode. So what happened? It spread across the globe. So the last is what is the conclusions? The Ebola outbreak in West Africa was a tragic event that exposed critical weakness in global and local health systems, and it caused the death of thousands of people. However, it also spurred unprecedented collaboration effort to address these shortcomings. One thing is disease is an opportunity, same time disease is a challenge also. The lesson learned from the crisis continue to shape global health policies and practices, emphasizing the need for a proactive and collaborative approach to prevent and respond to future pandemics. So when we talk about like that, we learn like that lots of, but same thing we observed during corona infections. We collaborated, some country developed a vaccine, some country produced a vaccine, some country dispatched vaccines through which we managed. So in India, we call this a vaccine diplomacy. We managed to give a free vaccine to many underdeveloped countries. Still, they remember about India mainly because of that vaccine diplomacy. The Ebola outbreak serves as a poignant reminder of the interconnectedness of global health. and imperative of collective action to safeguard and well-being of communities worldwide. So the local problem can be a global problem unless we have a coordinate effort to contain the disease from the place where it has originated. Thank you.